Welcome to Click for IB Biology. Here we're looking at gas exchange in the teleost fish, so we can contrast that with mammalian gas exchange like ourselves. Teleosts are what are called the bony fish, and they contrast with what are known as the cartilaginous fish. Cartilaginous fish are typically things like dogfish and sharks. We'll look at types of ventilation. We'll look at the buccal pump. We'll be looking at gill structure, which is this feature up here. And then we'll look at just some of the little details in parallel and counter current exchange systems, just very quickly. Should you wish to, in the description there's a PDF that you can download and annotate with your own notes as you go through the video. I will supply some key point summary, and there'll be a quiz where you can test your understanding of the video. So let's move on. So this is my attempt to draw a bony fish, a teleost. And in contrast, I've got a cartilaginous fish here. What you will know from observing something like a goldfish is the way in which the teleost fish can hold a stationary position, and they will open and close their mouths repeatedly, and this is part of their ventilation process. Contrastingly, something like a dogfish or a shark, they use what is called ram ventilation. And this means that the shark, the dogfish, has to keep swimming to force water through its mouth and across the gills here. The gills on a shark are seen from the outside as these vertical slits, usually about 5 on each side. In the teleost, they are covered by this structure which is called the operculum. So the shark needs to keep swimming, or it needs to hold position in a water current, in an ocean current, to maintain the ventilation across its gills. That is not the case for bony fish. What they do is that when they open their mouths, there's a negative pressure that sucks water into the space behind its mouth, which is known as the buccal cavity. Hence why we're calling this the buccal pump. Now the water then is pressurized, the mouth is closed, the water in this region is pressurized and forced across these structures here, which are the gill arches. The water gathers in what's called the branchial chamber, which is usually covered by the operculum, and then exits through this slit here in the side. By repeating this opening and closing of the mouth and pressurizing the water, the teleost fish is able to maintain the water flow over their gills. If we take a look at those gills, then we find that the surface area is increased on what are called the gill arches by these blades, otherwise known as gill filaments, and they occur in pairs as you go down the gill arches. And notice that the filaments of one fish of one gill arch overlap with the other. Now we should be aware that the water flow is coming from the back here, which is the buccal cavity, across the filaments, and then out into the branchial cavity before exiting through the operculum slit to the outside. And this flow is maintained by the buccal pump, which we previously discussed. Let's take a look at that for real. So in this image here, that is the gill filament, this time laid on its side. Here are the gill arches, rather. It's the gill arches. And these are the gill filaments. Here's another gill arch here. with the filaments like so. Behind is the buccal cavity, and this space in here, that would be the branchial cavity, and normally that would be covered by the operculum, which is here. When we examine a gill filament at the microscopic level, we find that the surface area to volume ratio has been increased not just by the presence of the multiple filaments, but they have vertical structures on them like so, which are known as gill lamellae. In this image here, this is the gill arch, these are the gill filaments, and so the lamellae would require further magnification before we could see them. Notice that the blood supply coming in is the afferent blood supply and the leaving blood supply is the efferent. In the gill lamellae, there are tiny capillary structures which allow the blood to flow from the afferent to the efferent. At the same time, let's remember that water flow is in this direction. So where we have water flowing this way, we have blood flowing this way, and this is called a counter current exchange. These gill lamellae, of course, are significantly adding to the surface area to volume ratio of the gas exchange surface of the gill. Just looking at the exchange system, so if our blood and water are flowing in the same direction, then we would have this set up here. So this would be the water here, and this would be the blood here. So if we consider oxygen, then oxygen would diffuse across until they come into equilibrium. And notice that that's not particularly efficient, that there's still oxygen left in the water which has not been extracted. With the countercurrent multiplier, so in this system here, we have water going in this direction and blood going in this direction. And so at each step, there's a small difference in the concentration, and so we would get a small amount of diffusion. However, if we take this across the entire surface of the gill, the diffusion will be much more efficient, and so we would get a small but accumulating diffusion of oxygen, and indeed in the opposite direction of carbon dioxide, making the countercurrent system much more efficient. And the countercurrent diffusion systems are very, very common in the highly predacious fish to support their high levels of metabolism as they hunt down their prey, demanding lots of oxygen for respiration in their muscle tissues.